Hello, my name is Javeria, and today I'll be presenting my summer research titled Common Ground, a cross-species brain atlas based on gene expression homology. To begin, the rationale behind this study stems from a major problem in translational neuroscience, where researchers often study mice to learn more about human disorders, but it's still hard to line up exactly where in the brain things happen. Our solution to this is instead of matching brain areas based on just their size or shape, we will use patterns of gene activity. Basically, we will align mouse and human brains by what the cells are doing individually, not just by how they look structurally. Through this method, we can produce a biologically meaningful tool that can assign a human equivalent region label to every point within a reference mouse brain. This will help bridge the gap of translatability within the field. For some background, since the primary goal of this project is producing an atlas, I would like to first define what an atlas is. A brain atlas is a collection of anatomical images where common sets of brain regions and their boundaries are defined. These images are typically put together to form a spatial 3D map of the brain. The schematic shown on the left is an example of how brain structures can be organized hierarchically in a human atlas, and the goal of this experiment is to map these brain regions onto the mouse. Essentially, I want to recreate a mouse atlas at different structure levels, much like how the human atlas is organized by levels already. In this project, I used the Allen Human Brain Atlas and Allen Mouse Brain Atlas, which are common databases used for thousands of studies to show which genes are turned on or off in each part of the brain. So how then did I do this mapping? I began by training a machine learning model called a random forest classifier, as shown right here, on human data. For each brain region, the classifier learned to predict whether human samples were from that region based on which genes were active, so which genes were on versus which were off. Once the classifier had been trained on all of the human samples, it was then applied to the mouse data, where it assigned a human equivalent region label to every point in the mouse brain. The outputs of the mouse predictions were region outlines as shown here, and confidence maps as shown here, along with a collection of human equivalent mouse brain atlases spanning from level 3 to level 9. To assess classifier performance, I used built-in classifier metrics to confirm that the model successfully learned human brain region labels based on which genes were active. However, some regions were more challenging than others to narrow down, and this is because biologically, some brain regions are just wired to do different things in mice and humans. For instance, the putamen shown on the left, which controls learning and motor functions, has been shown in previous studies to be similar across mice and human brains. And from my mapping, I found that the human definition of the putamen on the mouse brain looks very similar to a traditional definition of the mouse putamen. Conversely, the cerebellum varies a lot between species because of factors like it containing more white matter branches in humans as compared to mice, so the model had a harder time defining its boundaries, showing less accurate region matching and higher uncertainty. Finally, if there is one thing you should take away from my presentation, it is that I created a novel cross-species brain atlas. This atlas was organized hierarchically, reflecting the same anatomical tree structure shown earlier. The atlas displayed here represents the coarsest level, level 3. In the top row, each color corresponds to the boundaries of a specific brain region at that level, while the bottom row shows confidence values to assess how accurate each prediction is. In total, I plotted 269 brain regions across 7 atlas levels. In short, I produced a novel cross-species brain atlas based on how active genes were to explicitly define human equivalent brain regions on a mouse brain. This resource will enable direct comparison across species, advancing translational efforts. It will also be complemented by a corresponding human brain atlas that is being created by another student. Together, both atlases will be of particular interest to mouse imaging centers involved in translational research. Here are my references and acknowledgements. Thank you so much for listening to my presentation.